everyone, my name is Allie Denham, and today I'm going to talk about my year-long project on Skrjabingylus CHITWOODORUM, a parasitic nematode found in the nasal sinuses of Mephidus, commonly known here as the striped skunk in Texas. So, Skrjabingylus is a genus composed currently of 6 known species, 3 of which reside in the Americas, while the other 3 are primarily in Europe, Russia, or Asia. Skrjabingylus is known to target 2 known families of carnivores, such as the Mustelidae, which are weasels, badgers, or otters, or in this case, the Mephidids, which are the skunks. It has a really interesting life cycle, and for this case, they're all really similar in the life cycle, but we're going to focus just on the skunk right now with Skrjabingylus CHITWOODORUM. So it begins as a first stage larval phase in the fecal matter, and then from there it's ingested usually by the intermediate host, which is typically a gastropod or a mollusk, and then from there it's either ingested directly by the definitive host, which is the skunk, or it can be ingested by a paratenic host, which is, in this case, typically a mouse, or the mouse is ingested by a snake, which the snakes then are ingested by the skunk. And so once in the stomach, the larva is now a third stage larva, and then it burrows through the intestine into the abdominal wall and into the bloodstream, where it eventually finds cranial nerves and will travel up the cranial nerves to the olfactory nerves and then mature in the nasal sinus. As you can see from the photo that I have included in the poster, these parasites can get quite large, and there can be a lot found in one sinus cavity. I've read that up to 96 have been found in one sinus cavity, and so that definitely shows you how widespread and terrifying these little guys can be. So the basis of this project kind of began when I learned of a study of a former student who studied 500 skunks, and 48.5% had this parasite present in them. And I thought that was interesting compared to another study in Europe, particularly Germany, on pine martens, and only 1.2% had this parasite. So initially, this huge discrepancy was really interesting to me and intriguing, and I wanted to do a big genetic study on this parasite because no genetic work has been done on Skrjabingylus CHITWOODORUM, or very little work has been done, especially intraspecific variation studies. So that was the goal of this study, to do an intraspecific variation to see if there's any distinct lineages that are found, or if they simply do not show a high genetic distinction. And I decided that my main hypothesis would be that there was going to be a low genetic difference between them, and it was not going to have a distinct lineage. It was going to be just one lineage. And the reason why I chose this was because of a previous study done in Texas on the striped skunk that showed an incredibly high amount of gene flow, meaning that not a lot of variation was present. And so I figured the parasite was either going to do the same thing and also have low variation, or it would be different, and that's the alternative hypothesis, that we will see undescribed variation or barriers to gene flow and possibly even a new species, which is a win-win in this situation. So as you can see on my poster, I included the Texas map, and those are the sequences that we did acquire. The basis of this project focused on CO1, which is a mitochondrial gene, and we did a technique called barcoding, and it successfully tries to amplify just that one gene. It's typically known to be about 661 base pairs, and I would typically get around 530 for this project, and we successfully amplified 16 sequences, which are shown in the red, and it represents 14 different counties. And so I tried really hard to find different regions of Texas and get samples from them, and we made sure to have a very broad study. And as you can see, the blue dots are for my thesis I'm currently working on, which are the ones I've acquired for the thesis, and now we're up to about 28 counties, including some out-of-state ones, and up to 42 samples. So we chose CO1 because it was very variable and able to show variation and genetic differences between different genus, not species, but different genus between the huge nematode families. And so that's why we chose this initially. And so as you can see from my results, we created a phylogenetic tree to distinguish between the 2 known sequences of Skrjabingylus petraui and Skrjabingylus nasicola. As you can see, there is a low variation. All the CHITWOODORUMs did cluster together, which is what we wanted. As you can see by the bootstrap values, there's that 99, which means it's 99% confident that these are distinguishable from the 2 known other sequences, which is good. That's what we wanted. And then as you can see, there's very low variation, not many different lineages between the counties. And Hutchinson County, which is way up north, versus the B County one, way south, they're not so different after all. They're not very far apart when you look. And so that shows it's a low variation. I had an average of 0.8% variation, which is very low, but I had upwards of 2.5%, which is pretty high for a parasite. To distinguish Nasicola from Petrauri, it was 12.5%, so 2.5 is significant. And that was the whole basis, to distinguish them from the 2 known sequences. And I'm really happy with this project because we're now going to be able to add that 3rd sequence and help in the future endeavors and research of the 3 other species of Skrjabingylus that are not on GenBank. So thank you so much for your time and listening, and I hope you all stay healthy and safe, and thank you once again.